Hi everyone, my name is Fan, and the title of my talk is Comprehensive and Generalizable Prediction of Multiple Genomic Modalities. Recently, with the improvement of AI and machine learning, many models are proposed to predict different modalities of the human genome. This figure summarizes different categories of predictive models: sequence-based, epigenomic feature-based, and imputation-based. Sequence-based models predict features with DNA sequences. For example, DeepSeek predicts epigenomic features, and Akita predicts contact maps in some cell lines. The advantage of these models is they can reveal the effects of genetic variants on the predictive profiles. However, they cannot be generalized to unseen new cell types since all cell types share the same DNA sequences. Epigenomic feature-based models predict other modalities with epigenomic signals. For example, Caesar predicts chromosome contact maps with epigenomic features. These models are generalizable to unseen cell types, but relatively more inputs are required. There are also imputation-based models, which use existing profiles to predict missing profiles. For example, avocado uses available epigenomic features to predict missing ones. These methods are generalizable to new cell types with fewer available data, but it also requires retraining the model to include any new cell types. And a common issue of all the aforementioned models is they only predict one or two modalities. Therefore, we still lack a model for comprehensively predicting different genomic modalities altogether. And as I mentioned in the previous slides, many models are not generalizable to new cell types. They either cannot predict new cell types or requires extensive training to adapt new cell types. Then, the knowledge cannot be transferred across those predictive tasks. For example, the representation learned from DeepSeek cannot be used by avocado. And even though we have so many models, every task has to be trained and learned from scratch. Therefore, our goal is to develop a generalizable framework to comprehensively predict epigenome, chromatin organization, and transcriptome. Our work EPCOT is exactly designed for overcoming all these challenges. This is an overview of our model, just like the recent popular GPT or other related models which can convert the input text into the output text, an image, or a video. Our model uses DNA sequences and chromatin accessibility as an input and comprehensively predicts epigenome enhancer activity for the nucleome and transcriptome. There are a few reasons to include chromatin accessibility as part of the input. First, it makes the model generalizable for new cell types. It is usually cheap and available, and also it is also closely connected with other modalities. To handle all the predictive tasks together, we adapt the classic pre-training fine-tuning strategy. The large language model BERT also uses this strategy to achieve remarkable performance in many different tasks like question answering and machine translation. In BERT, the model is first trained with a massive amount of text data in two tasks: predicting masked words and predicting whether one sentence is next to another one. This pre-training stage helps the model learn the fundamental patterns of a language and the context. And then in the fine-tuning stage, the layers of the output part can be replaced by new task-specific layers. Then the new model can be trained with smaller datasets for specific tasks, such as question answering and named entity recognition, in which the new output layers are trained from scratch, but all previous input and embedding layers only need to be slightly updated or we say fine-tuned. So for EPCOT, in the pre-training stage, we train the model to predict 245 epigenomic features. Each input is the DNA sequences and chromatin accessibility of a 1.6 kilobases region, which includes the 1 kb center region and two 300 base pair flanking regions. The model takes the transformer structure. The encoder part uses self-attention to learn a sequence representation for the center region. The representation is fed into the decoder part, and to let the model learn the relationships and differences of these epigenomic features, we also input the label embeddings of all 245 epigenomic features. For example, the embedding of CTCF, ROT21, and H3K4 trimethylation. These embeddings are also updated during the training process, and in the end, it predicts the binary label bind or not on the center 1 kb region for each of the epigenomic features. In the fine-tuning stage, the decoder is replaced by task-specific downstream models, and this part needs to be trained from scratch, while the encoder part only needs to be fine-tuned. We defined 4 downstream tasks: epigenomic feature prediction, gene expression prediction, chromatin organization prediction, and enhancer activity prediction. These downstream tasks can be defined at different region lengths, referred to as receptive fields. For example, we could predict epigenomic feature for each 500 kilobases region, so the receptive field is 500 kb. And we choose these receptive fields just to match with the baseline method for better comparison with the baselines. In all these downstream tasks, we aggregate the sequence embeddings all over the receptive field and feed into the corresponding models for the downstream tasks. Here are some example outputs of EPCOT from cell line GM12878. We can compare the prediction results with the real experimental data, and this demonstrates that EPCOT can accurately predict gene expression, epigenomic features, and chromatin organization. Then we systematically evaluate EPCOT's performance in different approaches. First, we compare it with other models within cell type prediction or cross-cell type prediction results. And when we say within cell type, we mean training and testing on the same cell types but different genomic regions, which is an easier setting. And cross-cell type means training on some cell types and testing on other cell types. So here, we compare the RNA-seq prediction result of EPCOT and the baseline model attentive chrome, in which we do cross-cell type and the baseline does within cell type. We can achieve almost the same performance evaluated by the operation curve. We also find that when predicting epigenomic features, our prediction result is more accurate than the baseline method FactorNet evaluated by AUPR, which are both in the cross-cell type setting. These are just 2 examples, and you can find more evaluation results in our paper. Then we compare EPCOT's performance with imputation-based approaches such as avocado. In the epigenomic feature prediction task, we compare EPCOT with avocado in predicting 14 TFs in cell lines that are not included in the training set. 3 metrics, including AUPR and 2 different types of mean square errors, are used to measure the prediction accuracy on peak regions, and all of them shows EPCOT has a better performance. We also compare EPCOT's prediction performance with the average signal of all cell types to show it does not simply average everything. EPCOT outperforms average signal in all the downstream tasks such as predicting epigenomic features and predicting high-C contact maps. So after demonstrating the accuracy of our model, we have a new question. Currently in the fine-tuning stage, the representations from the input DNA sequences and chromatin accessibility data is fine-tuned in each of the downstream prediction tasks separately. However, we could also ask what will happen if we freeze the encoder part of the model and directly use the sequence representation to predict all downstream tasks. This is just like ask, can representations learned from embryonic stem cell epigenomic feature prediction be used to predict GM12878 contact maps? If we can do this well, it means the EPCOT model learns the knowledge that can be transferred across different predictive tasks. So we found that sequence representations from the frozen pre-training model were able to perform well in predicting gene expression and epigenomics, but not in predicting chromatin organization. In this task, fine-tuning significantly improved the performance in chromatin organization prediction. So this means in the pre-training stage, the model didn't learn some important features related to chromatin organization from the original input DNA sequences. Therefore, we adapt the pre-training model and then fine-tune the model in the chromatin organization prediction task for GM12878 high-C to let the encoder learn the additional information from the DNA sequences and chromatin accessibility, which is related to chromatin organization. we expect that now the encoder part can learn the relationships of the inputs with all other modalities and could produce generic or globally optimized sequence representations. And then in all downstream tasks, we freeze the encoder part, do not fine-tune it, and directly use the sequence representations as an input to check whether they can still perform well. So now we have two approaches. First, we can use globally optimized representations, which is pre-trained and fine-tuned in predicting chromatin organization and then frozen. And second, we can use the individually optimized or fine-tuned representations that are learned in different downstream tasks. We compare the two approaches in the performances of all downstream tasks. And here are the results. So in predicting epigenomic features, RNA-seq gene expression, cage-seq gene expression, micro-C, high-C, and enhanced activity, the globally optimized sequence representations achieved comparable performance with the fine-tuned individual sequence representations. And the model is also generalizable to other cell types other than GM12878. Therefore, it is safe to say these frozen sequence representations, which are yielded from fine-tuning with GM12878 chromatin organization prediction task, can be used as a generic or globally optimized representation of the human genome. This representation can be used for other few-shot or zero-shot training in the future. We also provide a web platform for every user to input their own data. On this page, you only need to choose a region of your interest, upload the ataxic data, and submit the job. The result will be ready in less than 1 minute. You can download them and visualize them on our web browser. That's the end of my talk, and this is additional information of our paper, code, and web server. Thank you so much for your attention.